Welcome, everyone. We are proud to introduce LungMap, and we have some big updates for everybody today. We are shifting our leadership at the 2-year mark, and so we have some introductions of our thank you to old people and introductions of new people. Dr. Borgai is not here today as our chair, who will be our past chair, and but he says thank you for everything and was with us yesterday. So we'll talk a little bit about our accomplishments. So moving into the future, and again, we're here to talk about what we're calling LungMap 3.0. You'll hear a lot about this, and it's really about how do we keep this platform relevant and moving into what will be the next generation of clinical trials. So this is from our meeting at ASCO and our retreat that we had earlier this year. Again, just to remind people where we came from, these ideas started back in 2012 and really opened a decade ago. So S1400 was the first master protocol for LungMap and included only squamous cell non-small cell lung cancer. Right on the heels of that opening, we had our first FDA approvals of immunotherapy. So this opened before immunotherapy was even a standard of care for patients. So quickly, LungMap pivoted and shifted into an era of refractory squamous cell and post-immunotherapy, and really was the first biomarker study, that prospective biomarker study for patients with squamous cell non-small cell lung cancer. Fast forward into 2019, we realized that we needed to keep up with the changing times, and LungMap expanded to all subtypes of lung cancer and continued to do prospective testing. And over the last 5 years, we've come to realize that that prospective testing was not as necessary as it was back in 2014. And many patients get NGS testing, fortunately, because of the advances in non-small cell lung cancer. And so we were actually doing somewhat of a disservice by requiring everybody to get repeat NGS testing. So we're still going to have an option for NGS testing for those patients who don't have it, and biopsy samples will be analyzed. But we'll also be allowing for patients to be using the comprehensive genomic testing tests that they already have and submitting those. And we'll be talking about that a little bit further. So we're calling this LungMap 3.0. Just a snippet of our current studies. And again, we still have biomarker-driven sub-studies. Right now we have 3 open, 1900E for KRAS G12C with sotorasib, which is quickly coming to its conclusion. We just opened S1900J at the end of September, and this is for patients with MET amplification, which is really a group of patients that we see frequently enough but don't have standard therapies. And exciting to say that this is the first non-industry sponsored amivantamab subcutaneous trial, so the subcutaneous version will be used. And then S1900K is utilizing tepotinib versus tepotinib and ramucirumab for exon 14, MET exon 14 skipping. We have our first study in molecularly identified but resistant tumors, and S1900G is specifically for EGFR patients with MET amplification, with capmatinib, osimertinib, plus or minus ramucirumab. And then we're working on another study, S1900N, looking at TROP2 plus or minus PD-1 VEGF. And then we always like to have our non-match studies when we're hoping S1900E will be opening in the next few months. We're always trying to, again, continue to move this forward, to continue to be inclusive. And so we have some initiatives for the next number of years. And we're going to be increasing engagement of junior investigators, establishing mentorship plans. We're trying to work more closely with our translational medicine subcommittee during the trial development and afterwards to try and utilize these samples that we have and get junior investigators involved in some of these studies. We want to look at novel immunotherapy combinations and biomarkers and potentially biomarker-driven sub-studies in the immunotherapy setting. We want to strengthen our relationships with our industry sponsors and help to highlight the benefits of working with LungMap, one of which Mary and her team highlighted last year, where we see that with our diverse NCTN network, we increase diversity in our population and have more rural sites than most other, more rural accrual than most other studies. And we're starting to see this in our the spin-off of S1800A, the Pragmatica lung study, where the diversity is probably the most significant I've seen in any study. And we continue to work with our patient advocates and DEI to increase trial representative, and often this is at the site level. And I want to do a shout out to Lucy Ganzauer, who is our fearless champion in DEI, and really what she accomplishes in a day probably is what I can do in a year. So we're going to move forward, and we want to focus our energy at building something new, but on this amazing platform that has been around for now a decade. Thank you.
Yeah, thanks, Karen. I mean, it's it's been 10 years now that LungMap has been open, and so it's exciting to see all of these changes. So I'll just briefly give you an update on where we are in terms of manuscripts and accrual. But I want to note first that we're anticipating that the screening accrual to LungMap is going to change dramatically with this new version of LungMap. David Kozno is going to go through the details about how patients come on to the study, but I think you'll see why I mean that. So from the start of LungMap, we've required Foundation 1 testing which has required submission of tumor tissue to the study. About 3 years ago, a little bit more than 3 years ago, we outed the ability for patients who had already had the Foundation 1 CDX test completed that they could use that to come onto the trial. But moving forward, patients are going to be able to use their NGS test results if they've already had those completed. So since LungMap, which is the protocol that opened in January of 2019, so that's LungMap all caps, we have a total of 3,512 patients that have enrolled to the study. About a third of them have been patients who have been screened at progression, and 2/3 of them who are patients who have been pre-screened. And of those, 313 have been enrolled in the last year. And since we implemented the allowance of using the reanalysis, the previous Foundation 1 tests, about a third of the patients who are coming onto the study have been using that option. In terms of patients who have been assigned to a sub-study, so remember you get screened, and then if you're screened at progression, you just get assigned to a study if there's one available right away. If you're pre-screened, you need to have progressed on your current line of therapy and then submit a form to tell us that that's happened. And so in terms of the 3,512 patients that have been enrolled to LungMap, 1,697 have been assigned to a sub-study, and 58 of them have been assigned to a study in the last year. In terms of total sub-study accruals, we've had 490 patients who have been registered to a sub-study since January of 2019, and there have been 26 of them that have been registered to a sub-study in the last year. Now, noting that while we do have currently 4 biomarker-driven studies that are open to accrual, about 2 of them have just been opened very recently, and so we're anticipating that sub-study accrual is going to increase, but we also think that sub-study accrual is going to increase because of the changes that you're going to hear about in a second. So in terms of manuscripts, we don't have a lot to report. We have a lot of things in the works. In the last year, we've had 1 major publication that has come out, and I just want to congratulate David Kozno on this paper. So what we did was we looked at the next-gen sequencing data from the first LungMap study, S1400, that looked at only squamous cell patients, and we evaluated if there were any genes that either were co-occurring or mutually exclusive, and we found some pretty interesting results. And so if you're interested in seeing that paper, you should go to JTO and look at a paper that has David Kozno as the first author and I think me as the senior author. So it was a pretty interesting finding and just a nice way to show that this is the largest set of squamous cell data I think that's out there, in particular in the second line setting. We have some other pending manuscript updates coming out. We have a couple sub-studies that have been completed a while ago and presented at ASCO. So S1900A was a study looking at a PARP inhibitor. Patients were enrolled into cohorts by squamous and non-squamous cell histology. So that paper is almost completed and ready to be submitted. And then we have a study S1900C, which evaluated avelumab with talazoparib in patients with STK11 mutations. And that study paper is nearly completion, and so we'll be trying to submit that soon. We have some translational medicine analyses that are pending as well, and so pretty soon a paper that's comparing detection of gene alterations in ctDNA with tissue will be submitted to a manuscript, to a journal. And in addition to having some interesting analyses, I think one of the major import of this analysis is that this is a data set of patients in the second or later lines of therapy evaluating not just the co-occurrence of detection in both ctDNA and tissue, but also just the prevalence of these gene alterations, because it's really, I think, one of the only data sets looking at this population explicitly. We also have some analyses that we're looking at in terms of ctDNA in the S1800A study that Karen just talked about. That was pembrolizumab versus standard of care in the second line or previously treated immunotherapy setting. And then there's another analysis that's looking at just KRAS alterations and evaluating how they do in different settings to try to understand that in more full. So there's a lot going on, and I think that hopefully in the next year, when we meet again, we can give you an update about some more papers that have been published. Thank you.
All right, so it's my true pleasure to announce that LungMap 3.0 officially started on the 15th, just this week. And so here, I'm going to tell you a little bit about why we're doing this and how it's going to help find patients for our studies. And so Karen and Mary both got to this, but back in 2014, when LungMap started, it provided a real service for the patients, which is at the time had squamous cell cancers, and molecular testing for these tumors was not at all standard of care. There wasn't really an avenue for people to get next generation sequencing for patients with these tumors, and this was just a decade ago. And so it provided access, and it allowed a whole battery of sub-studies to be run based on the results of these screening tests that identified mutations and other alterations that would guide patients onto the right drugs based on those results. But fast forward here to 2024. Screening proved itself. We've an increasing arsenal of new drugs that target specific alterations seen in lung cancers, both squamous and now non-squamous. And patients have, because of this, been increasingly able to get NGS testing on not just one or two, but a whole slew of different tests, both commercially developed and at a variety of different academic hospital settings. And this is what we've been increasingly hearing. We know this patient has this KRAS mutation, but it's not from a Foundation Medicine test. Do we really have to find the tissue, maybe even discover there isn't enough for retesting, send that in, wait 16 days up to for a result, and then only put a patient on a trial for this recurrent or progressive disease that they really want to start treatment for yesterday? And until we've made the allowances in some of our sub-studies to try to help with that, we've had to continue on this process, but no longer with this change. And so now we want for any CLIA certified test that meets criteria as being what we call sufficiently broad to inform lung cancer biology, to look at lung cancer genes that we know are important in lung cancer or are deeply interested in studying. This is the list that we currently have today. I should mention this is going to be a list that is likely to evolve with time as we continue to understand what drives lung cancer biology. But for now, if you have a gene panel that gives yes or no answers to the presence of an alteration for each of these genes, it's going to meet muster for our study. Typically, this is going to be an oncology-focused panel of about 150 or more genes. It pretty much splits out this way. If the panel has, let's say, under 100 genes, chances are it's going to miss one or two of these genes. But if it has 150 or more, virtually all of the panels have all of these genes. So it's actually a good number of panels that already meet this definition. And then beyond just even being able to tell us about these genes, there would be enough data to calculate things like tumor mutational burden, which we know is important in immunotherapy, which itself is super important in lung cancer. So what we wanted to do is to be able to have a list of tests that we know would be good for lung map. But we also, as I'll speak to, wanted to have a process where we allowed any test that would meet these criteria to inform patient eligibility. So as a starting point, so as not to completely reinvent the wheel, we started by looking at efforts like NCI Match, which 2 years ago closed to accrual after enrolling over 1,200 patients to 38 different treatment arms for different solid tumors and liquid tumors. And they had this list of commercial and academic labs, which is the same kind of consideration we've had today, although not everyone on this list ended up meeting our decided upon criteria, so you'll see the lists are a little different. But they did a rigorous process back in informing NCI Match, even doing a concordance study, looking at cell lines and clinical samples to see what the concordance of the results of these tests were with the central test. But given the maturity of the NGS testing since then, it seems that this extra step isn't always going to be as needed as it once was. And so here's the current list. For commercial tests, each of the ones listed here meet the criteria of having all the genes that we need to inform study entry into LungMap. There are specific companies and specific tests from these companies, but this is, again, not meant to be an all-inclusive list. It's everything our group tried to think through and looked at. There are a few maybe common ones out there that aren't going to be on this list, and we're going to actually try to include that in a list as well so people don't have to wonder about those. But this is the starting list for commercial tests, and this is the starting list for academic lab tests. So you can see there are about 36 already that we know are pre-qualified for LungMap. So if a patient had an NGS test from any of these sites or companies, you're good to go. Now, tumor testing, it's been the gold standard, but increasingly we found that we can identify the tumor DNA in circulation in the blood, ctDNA, and there are 3 companies whose tests currently have enough different genes looked at that they also meet our requirement, and they are these 3, as you see listed here. But again, our hope is that this list grows over time as tests reach the criteria for LungMap. So to identify what are the pre-approved lists, I showed what we have as of today, but this is going to be a living list. As companies create new tests that meet muster, we're going to add them to this reference page. It's going to be this website that you can always access, and then you can check to see if a test is or is not approved. If it's not on the list at all, it may just simply be that we haven't encountered it before in LungMap, so we want a chance to see if it might work for the program. And so all you'd have to do is send the de-identified test to this email address, and we're going to try to qualify it ASAP. Someone actually already just sent one of these yesterday, and I tried to answer right away since it's one I've already looked at. But the idea is a group of us is going to be able to staff this so we could try to do this hopefully within 24 hours, so you'll have your answer right away about if a test can be used. And then you'll put the data, mostly yes or no, is a certain alteration for the key genes of interest present or absent. There is a field for unknown, but hopefully that's not going to be a common entry because the idea was these tests do test all of these genes. But we're going to have this genomic alterations form that you could see right now at CTSU. And then this is how we're going to be able to collect some structured data.
Can I interrupt for a second?
Oh, please.
It sounds like somebody online is saying that they've lost audio.
Wait, what?
They said they've lost, no, it wouldn't be, is it,
not-
are you, we hear you, it's
Yeah,
not you.
okay.
They're saying online that they might have lost audio?
No, we can
They
still
can still
hear.
hear. Okay, thank you. I just wanted to make certain.
Oh, yes. Thank you, Mary. All right. So this flow chart that Mary and her team have been preparing is super important. It looks a little complicated at first, but the whole idea is to keep all that's been good in LungMap and yet enable all of these new ways into the study that could hopefully greatly help identify patients. And so in the way she's laid this out nicely, on the left is essentially what we've done to date. So nothing really changes about that. You absolutely can still send tissue in for testing. It would be done by Foundation Medicine, F1CDX. It can be done at the time of progression, as we have, and it could even still be done at pre-screening, which means before the patient gets progressive disease, but so that you can do that testing, with some time to spare and get results that would be then ready to go if and when they should have progressive disease. So this is still, I think, a great feature, and as I'll get to, there'll be even other good reasons for maybe wanting to do so as we build more things into the central testing. But again, nothing here changes. So that's one bit of news. Everything that did change, though, is on the right side of the line here. And it's really about how we use all of these prior test results. And so you'll check to see if a test is on the list of pre-approved tests. And if it is, great. Then we just need to know if it's from a tissue test or a blood test. If it's a tissue test, you would then fill out the results on the case report form, this genomics alteration form. And then within a day, you should be able to get sub-study assignments. So this is meant to be a very quick process. It should be really helpful for the patients. Now, one caveat, if it's a blood test, we do need some way of knowing that the test is actually picking up tumor DNA in blood. And there are a lot of good bits of investigation underway for creating criteria to say, what is a blood test that's successfully picking up tumor? Because even with stage 4 disease, you don't always get enough tumor DNA shed into the blood to be able to reliably say if something is present or absent as an alteration. But we figure that if there is a tumor-specific alteration, say like a TP53 mutation seen that's picked up on a blood test, even if we don't have a sub-study that's specifically looking at a TP53 drug, we don't. But even if we don't have the sub-study, the fact that alteration was seen in the blood tells us the test is picking up tumor DNA in blood. And so then we know it's there or not, we could trust the results, and then we could use it the same way as tissue. However, if not a single gene is picked up on our list of genes, then until we have additional criteria to say the blood test was able to make a call, we would still need tumor tissue testing in that case. If a test is not on the pre-approved list, the only additional step is that you send it to that email address, and we'll try to say right away, yes or no, it can be used for the study. So then, okay, LungMap's been from the beginning about creating new opportunities, understanding lung cancer better so that we can then create new sub-studies, test new therapeutics. So we want to keep on being able to do it. And the piece that we have ready for here in LungMap 3.0 is now bringing RNA testing as part of the standard package of what will be done for any tumor tissue that's sent into LungMap. And this is going to be using the Foundation 1 test. It's a research use only RUO test at the present time, but it's going to be able to look at rearrangements in 318 genes and gene expression levels in over 1,500 genes. And a couple good points to make about this is that sometimes it's much easier to see a gene rearrangement or fusion in RNA compared to in DNA. There's good research along these lines. And so, because there are a number of drugs that are based on identification of rearrangements and fusions, this would be a good reason to get RNA testing, and this will be now provided on LungMap. So this is fantastic. And then what we can learn about gene expression, I think could be great for correlative science and to really inform the next set of studies because there's a lot of biology that does not get captured on the DNA genetic level, but only things that are past the DNA level, including RNA. And so I think it'll still be very useful for tumor tissue to be sent in for testing on LungMap. That said, we still want to be able to find patients today for the studies we have active now. And so I am very hopeful that this new ability to use any good CLIA certified test is going to help find patients for our studies.
So there's a question online. So where do we find this genomics alteration form? And well, that is specified in the protocol. It's part of the lung map protocol. And so it's in Medidata Rave. You'll see the master form set on CTSU, but it's all just part of the protocol, and you'll see all of that information.
Yes, and then, so I didn't have the chance to put it into this slide deck, but I do also want to allay concerns, because for those folks who have seen some of these commercial and academic lab reports that list all of the different alterations seen or not seen, it can seem a little daunting at first if you're not used to looking at these. And so we've been preparing help guides that basically mirror what's in the genomics alteration form, give the result based on our interpretation of the report, and highlighted the reports themselves for where you'd find all the information, and then even said in a column what page it's on, what header to look under, for several of these tests. And we're going to continue to try to update and flesh this out over time. And so hopefully people can use these as examples as you then see, the reports you get for your patients. If in doubt, there are still questions, we want to be available. I definitely want to be able to help make sure that we get the right information so we get the patients on the right sub-studies. So if you want to reach out to me about any comments, concerns, worries, here's my email address.
David, could you comment on what impact you think that is going to have on the screening accrual, but also the success rate of patients screened to go on to sub-studies?
100%. So, I think, Mary, you said it's about 1 in 5 or so patients who ultimately end up on a therapeutic sub-study.
Yeah, although pre-screened patients, it's smaller than that.
Yeah. So, one thing about the way we've done things, because, the whole initial structure of LungMap was we needed to screen for alterations, and you don't know if you'll find one for a sub-study. We've been able to address this in some part with the non-matched studies where patients could go on when they don't have any of the applicable alterations, but it's still a relatively small percentage of patients who go through all of that to participate on the screening study and then never end up on a trial that's meant to try to help them. And this is a shame, and it's work, and and patients are waiting, to hear hopefully good news, and maybe they don't. What's different here is that you already know, because you already have a result of one of these NGS tests, that, hey, they have that, KRAS mutation, for example. Then you just can say, oh, and LungMap has a study against, that particular alteration. Since you already know that they could participate on the sub-study, we're hopeful that the percentage of people who go through this screening process, which for prior results would literally be just submission of the paperwork, will go on to a sub-study. So this could drive, several fold potentially, the percentage of patients who get screened and then end up on a sub-study that we know they'd be good for. Yeah, and I should just, if there are any concerns about concordance, these days it's vanishingly rare for one test to show a mutation and not for another test to pick up the same one. So, we feel confident that we can use all of these tests and still get patients onto the right studies. Great, thank you.
Thank you. And we'll call up Frank DeSanto for our Accrual Enhancement Committee updates.
Thank you for the opportunity to present on behalf of my Accrual Enhancement Committee colleagues. What we've been focused on since the last group meeting has been primarily achieving the priorities set in the LungMap March retreat, some of which are identified on the slide, and I'll dive more deeply into them. One of them is getting out a message to cancer centers that they should be opening all of our sub-studies, and that the NCTN RFAs, the NCI RFAs, allow for exceptions in terms of counting accrual for rare tumors and molecular phenotypes. Master protocols should be tracked collectively for grant reviews. So we worked with leadership to draft up a message, and we're assuming we would have to send it out maybe under Dr. Blanke's name. And then we talked about the other precision medicine protocols that the NCI has going, maybe facing similar challenges. We reached out to MyeloMatch, and they're concerned about the same thing, and let us know that actually Combo Match is already facing the problem, and they were, in fact, working on drafting a similar kind of message. So we talked about broadening it so that it could cover all master protocols. And right now, it looks like that message is going through review at NCI Division of Cancer Treatment and Diagnosis Leadership, and will go out from them to Cancer Center leadership at some point soon. We also worked to develop messaging for the various audiences we want to reach about the revision, LungMap 3.0. And these are messages that we've used and will use to populate our promotional educational documents. This includes more messaging and outreach for industry partners, including focusing on the benefits of partnering with LungMap. And leadership has developed a pretty extensive list of benefits, which we will be rolling out on the lungmap.org website soon as well. In terms of outreach to sites, in addition to our regular protocol updates, we've got the LungMap newsletter, which goes out to active site PIs and primary contacts. And there's an issue that will go out soon. Of course, it will highlight the big revision, LungMap 3.0, along with the message that what drove this revision was really feedback from sites. This is what sites have been asking for some time. But since S1900J just launched, we'll talk about that as well. And just mention that soon we'll have a new non-match sub-study as well. The training team has rolled out a new site initiation training webinar in CTSU's learning management system. We'll be presenting LungMap on the monthly NCORP admin webinar, I think later this month, actually. And that will include some targeted messaging for community sites specifically. We've sent out to our study champions a LungMap 3.0 slide set that they can use in, for example, presenting on the trial revision to other group meetings. One of the venues for the messaging we developed is, of course, social media. And we've started that campaign already. In addition to those messages we developed, we also reached out and continue to reach out to leaders in a number of areas, including leaders of advocacy organizations, NCI leadership, obviously LungMap leadership, to get statements of support for the trial, and are including those in our outreach on social media. So if you follow @LungMap on Twitter, you've seen these and we'll see lots more of them. We're also working on updating the lungmap.org website, which includes just refreshing of some messaging, but also including the LungMap 3.0 messaging, and that will be rolled out very soon. That's ready to go. And we've mocked up an industry partners section as well, incorporating the list of benefits for industry partners that I referred to a minute ago. That will be rolled out soon. Yet to come, we'll be focusing on outreach to advocacy organizations. Last summer, we hosted a webinar for advocacy organizations, was very well attended, and it's actually been watched quite a few times since then, the recording of it. And we'll be doing something similar related to LungMap 3.0 in the coming months, probably reusing some of that previous webinar content in modular fashion. The social media push will obviously continue throughout the fall and beyond. But once most sites have had time to get the revision going and start implementing those processes, we will also do a press release and a publicity push around that. That will be later this fall. And we're working with leadership on an editorial to be submitted to a venue like the ASCO Post or the Cancer Letter to try to reach the cancer research community with messaging about the revision of the trial. Last point, just a pitch for the resources we make available for all sub-studies, social media toolkits, but particularly the clinical trial summaries, the patient-friendly summaries, always available at swog.org/S1900G, S1900K, et cetera. Thank you.
Thank you, Frank, and we'll bring up Lucy Ganzauer to talk about representativeness.
Thank you. I just want to do a quick update. One of the first things I want to call your attention to is LungMap has really led in ensuring that we have representativeness and more real-world populations. But like everything else with COVID, we had sites that were short on staff and weren't opening all the arms. It is really critical that we have real-world populations and the entire portfolio with all the arms are open at your sites. This is an amazing umbrella trial that offers really cutting-edge treatment for your patients, and having that entire umbrella open is really critical. This trial works really well in small sites. A small rural hospital in South Carolina, ANMED, has been in the top 10 on accruals. So if it can work at ANMED, it can work anywhere. We also can offer doing webinars to your community investigators. We know that very few of your community investigators can attend these national meetings, but through the Community Engagement Committee, we can do one-on-one webinars with your staff, your coordinators, or with your investigators to get their buy-in. One of the things that's coming out that I just wanted to make people aware we'll start seeing changes is the OMB New Directive 15. This was released in March of this year, and it's going to start having just one question on race and ethnicity, which really makes sense and is going to be a lot easier for people to fill out accurately. And they're also adding a new category, Middle Eastern or North African. And so there's going to be more information on this coming out, but anything that has to do with the feds has to put in an action plan by September of next year. So we will probably be seeing that in grant applications and other things. These are, and also a report that came out from the OIG that really kind of criticized NCI and others on our trials that we often fall short of underrepresented groups. We do know that when we put our effort to it, and SWOG has made a huge effort on trying to ensure representativeness, and our pragmatic trials shows when you put that effort and you follow guidelines on this and monitor it, like our committee has been monitoring it in multiple committees monthly, that those results pay off and we can have our trials very representative. And SWOG, because of its large network, and particularly of the in-core sites, where over 50% of our accruals come from, we can really deliver on that. So this is just showing some of the critical statements of showing that most of the trials did not have an inclusive plan and that they missed their targets. And so we really can do better. And so this is showing that OIG recommends that the NIH hold researchers accountable. They want us to develop inclusive targets and that we're to start having the new demographic data. So we will see those things coming forward. So this is a new minimum data set They have a larger data set as this, but this will be the new minimum data set coming forward. This is the FDA guidance on diversity plans, and that was really the roadmap that we used when we were designing the pragmatic. And there's a better article that really goes through this model diversity action plan. If you haven't looked at that, I recommend it. It really gives kind of less federal language to meeting those and good examples. It takes a village, and real-world populations come from your communities and your outlying clinics. And so I want to continue to advocate for your voice to help us make sure that our trials are diverse. Thank you.
And we have online, Dr. Pata.
Hi, Dr. Reckamp. Hi, everyone.
Hello. We don't see you yet.
Okay.
Oh, maybe, do I have to click? Oh. Great, okay. So we'll advance your slides and we'll start here.
Fantastic. It's my pleasure to be with you. I'm going to give you an update today on S1900E, as you heard from Dr. Reckamp, which is a study examining sotorasib in patients with KRAS G12C lung cancer, really examining the impact of co-mutations for this drug's efficacy. So I'm presenting on behalf of my co-chair, Dr. Gerber, as well as the lung committee chair, Dr. Brahmer, our ECOG study champion, Dr. Neal, and then our wonderful lead statistician, Dr. Redmond. Next slide, please. Many of us are aware that we have exciting development in KRAS G12C lung cancer and those previously treated with these KRAS G12C allosteric inhibitors with sotorasib, which underwent accelerated approval in May 2021. Subsequently, adagrasib with approval in December of 2022, based on the efficacy from large phase 2 expansion cohorts, which demonstrated response rates of around 40% and median progression-free survival around 6 to 7 months. Next slide, please. So after that accelerated approval, we now have heard data from 2 confirmatory phase 3 studies with both of these drugs, CodeBreak 200 with sotorasib and Crystal 12 with adagrasib. Both of these were randomized studies that randomized to our previous standard of care, docetaxel. There were some slight differences in study design, but I think both well-designed studies with a randomized component. So next slide, please. So what do we see in terms of progression-free survival and other efficacy standpoints with both of these drugs? It definitely appears to be better than docetaxel as it relates to the primary endpoint of both studies of progression-free survival, with very similar hazard ratios, improved response rate, median duration response compared to docetaxel. Now we know from CodeBreak 200, where there was a lot of discussion around this, that the median overall survival was no different actually from docetaxel, and there was some crossover in that study. For Crystal 12 for adagrasib, we're still waiting for overall survival data, but I think, you know, we have a long way to go for KRAS G12C lung cancer, but obviously this is improvement for these patients with this disease. Next slide, please. So we know KRAS lung cancer is also quite heterogeneous. One of the ways that it is based on additional co-mutations that may be detected in broader next generation sequencing panels. You can click through the animation. I think there's one more. Okay, perfect. Okay. So both sotorasib and adagrasib have demonstrated potentially that STK11 co-mutations may not have impact on the response rates of these drugs the way we hypothesize that they do for immunotherapy-based treatments, for example, while those patients with co-mutations in KEAP1 may have inferior response rates with both of these drugs. But again, these are retrospective looks of prospective studies. It wasn't really designed to answer this question. Next slide, please. So it gets a little bit more complicated. So we were just looking at response rate there, but maybe it doesn't matter so much in terms of co-mutations as it relates to progression-free survival. This is a subgroup analysis from the sotorasib phase 3 study where patients with P53, STK11, and KEAP1 alterations all derived benefit from sotorasib. So it's a little bit complex of a story. So next slide, please. So that's why this trial has been designed. In fact, this trial launched just before the sotorasib accelerated approval. So it's kind of amazing that we are almost fully accrued. So these are patients with recurrent KRAS G12C lung cancer who have received at least one prior therapy. And these tumor samples have undergone rigorous biomarker testing to define the co-mutation cohorts of P53 and STK11, but we also wanted to make sure we had a catch-all cohort to allow other patients to enroll on the study. So as you can see, the P53 mutation cohort is fully accrued. Congratulations. The STK11 mutation cohort as well, fully accrued. Our catch-all cohort only has about 4 slots remaining, but I'll tell you what we're going to do about that because this has been a relatively slower accruing cohort. Our primary endpoint is objective response rate, so we'll get an early readout on efficacy, but we'll of course also look at duration of response, PFS, OS, and all of these different co-mutation subgroups. Next slide, please. So I said we have slots available in the all other co-mutation cohort. However, we just did submit a protocol amendment to change our available sample size from 40 to 36 to be able to shortly close this study to accrual so we can start to begin to analyze our data once patients have undergone enough follow-up time. As I mentioned before, I think this will be really give us a clear signal, at least in terms of how co-mutations impact the efficacy of these drugs because of how strict the co-mutation definitions were in the study. And then as you've heard from many speakers already, we're really hoping to represent a more racially, ethnically diverse patient population, the real world population when we're treating patients in a variety of communities. So at 95 participants, you can see the race ethnicity breakdown there. We, I think, are doing a decent job. Obviously, can always do better, but we also tried to do as best as we can with this very strict biomarker definition to try to make other things easier in the protocol. I think that's a good take-home message for all of us in future design of studies. I thank you for your time.
Thank you, Suki. Our contacts are here. We'll move on to S1900G with Dr. Goldberg.
Thank you very much. Hi, everyone. So I will be talking about S1900G, which is for patients with EGFR mutant lung cancer with MET amplification. So I showed this yesterday at one of our meetings, so apologies for those of you who've seen it. But I really wanted to talk, before talking about the specific sub-study, is about how we think about treating patients with EGFR mutant lung cancer. Things have changed over the last couple of months even, so it's become a complicated situation. So patients who are newly diagnosed with metastatic EGFR mutant lung cancer typically receive an EGFR inhibitor in some capacity as their first-line treatment. It used to be osimertinib on its own. Now it's become more complicated, and sometimes it's in combination with other agents. Typically, the treatment works as first line, and tumors shrink and patients do well for a period of time, but all patients eventually progress. And so typically, osimertinib or another EGFR inhibitor is continued in some way as part of the treatment, and then disease will progress. So what happens at the time of progression? Standard of care, for the most part, I would say most people would agree that it's to get molecular testing and try to understand the characteristics of the tumor. And so you can do that in 2 ways. You've already heard a lot about this already at this meeting. One is through blood, looking at ctDNA. Another is doing a tumor biopsy. And in clinical practice, a lot of times we'll do both. Sometimes we'll do blood first, and then we'll do a tumor biopsy if blood doesn't show anything. Sometimes we'll do biopsy upfront. It varies, but often we will have one or both. And so what happens at the time of progression on osimertinib? A whole variety of things could happen.
And that's why it's so important to look. One of the things that is more common than others, although still a subset of patients, is MET amplification. So this is something that happens in the tumor only at the time of progression. It's not there up front in the vast majority of patients. So you can only find it if you do a blood sample or a biopsy at the time of progression. And that's the focus of this sub-study. So this is what I basically just said in words, that patients initially do well, but then they progress, and MET amplification is found at maybe 15 to 20% of patients progressing on osimertinib. Based on a couple of other studies that combining a MET inhibitor with an EGFR inhibitor at that time of resistance when patients have MET amplification can be effective. This study I'm just pulling is one such example where the response rate was 30% and the PFS was 5.4 months. And because it's relevant for this study, capmatinib is already an FDA-approved drug, but it's used for patients with MET exon 14 skipping mutations. But it is a MET inhibitor that we're using in this trial. And so the other part of this trial is looking at VEGF inhibition. So we know a lot about VEGF inhibitors in EGFR mutant lung cancer. We know in the first line setting in several studies that it can prolong the progression-free survival in patients with EGFR mutant disease. And this is, again, just one such example, which is looking at osimertinib with or without ramucirumab, where the PFS is improved at 24.8 months versus 15.6 months. Again, this is the first line setting. So what is this trial looking at? Well, it's looking at patients with EGFR mutant lung cancer who have received osimertinib as part of their most recent line of treatment. It doesn't have to be immediately following first line. They could have had other treatments, and it could have been given in combination. But osimertinib must be one of the most recent drugs that have been given. And these are patients who all have MET amplification, either from blood or tumor biopsy. They can have had a prior MET tyrosine kinase inhibitor, although the drug amivantamab, which is an antibody, is allowed. And they can have had prior VEGF pathway inhibitors. Those are two changes that have been made in the recent couple of months with a prior amendment, allowing prior amivantamab and prior VEGF pathway inhibition. And untreated asymptomatic brain metastases are allowed. And so these patients are randomized to the triplet of osimertinib with capmatinib, the MET inhibitor, with or without ramucirumab. And the primary endpoint is progression-free survival. So you've already heard a lot about biomarker identification on LungMap. This used to be somewhat unique to this sub-study, and now it's not, because now, fortunately, all of LungMap has evolved. But this is just to highlight that, again, the EGFR mutation will be found at baseline in these patients, and MET amplification will be something that's at progression on osimertinib. And again, you can find it in the blood or in the tissue, and it could be central testing through LungMap or local testing. So here's our updates. So the accrual goal is 66 patients, for which we imagine 60 will be eligible. Our hope was to have 2 patients a month. It opened in April of last year. We actually have 16 patients registered. So in the last couple of days since I updated these slides, we've had one more. So we have 16 patients. So we've had somewhat slow and steady accrual. So thank you all for thinking about this study and putting patients on. It has been somewhat slower than we expected, but it's been continuous. So keep up the good work, keep looking for MET amplification. You won't know if someone has it unless you test. And hopefully we can complete this study in the next year or 2. I will put a plug in for this educational webinar that we're planning. We don't have a date yet, but it's going to be sometime in early 2025. Because of the evolving landscape in EGFR mutant lung cancer, we thought we would have an educational webinar about EGFR mutations in general, the first line treatment, second line treatment, how we think about resistance, and then focus in on this sub-study, as well as maybe some other, we had a great suggestion yesterday about maybe focusing on all of the EGFR studies throughout the cooperative groups and really trying to put those into context by talking about EGFR disease in general. So I think that's all I have. Thank you so much.
And we will have another online discussion for S1900K with Dr. Peck.
Hi, everyone. Hopefully the audio is working. And to continue the theme of MET, I'll be talking about S1900K, which is taking a look at MET exon 14 skipping as a primary driver in a randomized phase 2 study of tepotinib with or without ramucirumab in second line, and I'll get to that in a little bit, and beyond non-small cell lung cancer. I'm presenting on behalf of my co-chair, Janine Lee, and of course, our excellent other study team members who are listed there. Next slide. So as way of review, I'm sure everyone's familiar at this point, MET alterations are relatively new still as a interesting target, particularly as it relates to exon 14 skipping, which happens in about 3 to 4% of all non-small cell lung cancer cases. These involve mutations in the flanking intronic sequences surrounding exon 14, which leads to alternative splicing, in-frame fusion of 13 and 15 following deletion of 14 in mRNA. And evidently, this causes alterations in the juxtamembrane domain, which prevents civil binding and ubiquitination. So you get target stabilization, which evidently is sufficient for oncogene detection that we can target. Next slide. So we do know now at this point for tepotinib and capmatinib in the US and a number of other MET TKIs that have been approved in China, that it's a generally effective therapy. Overall, if you were to just squash all of the data together, you get overall response rates about 50% and a median PFS of between 11 to 12 months, and that's shown in the purple dot there. The issue, of course, and I don't know if this animation is going to work. Try next slide. There we go. But the issue, of course, is that we're used to much better results for other targets and their associated drugs, and that would be the case pretty much for every next-gen ALK inhibitor. Some hits right off the bat were crizotinib in ROS1-positive lung cancer, and osimertinib also in EGFR mutant lung cancer. So our target generally at this point is response rates between 70 to 80% and a median PFS in excess of 18 months. And so with that benchmark present, there's some thought that we're missing something in the upfront setting. Next slide. One of the other things that we have realized is that the side effect signature for MET inhibitors is a little unusual and ultimately unique in the therapeutic world, marked by fairly high rates of peripheral edema. So most patients who are treated with a selective MET inhibitor will end up getting peripheral edema. In the pivotal trials, rates of peripheral edema are generally around 60%. Most of these, we say, are grade 1 or 2, but a grade 2 peripheral edema event is still pretty important and something that can impede quality of life in an elderly population. This is associated with other vascular permeability issues that we do see, hypoalbuminemia, which can make the edema worse. We see some creatinine processing issues that occur, as well as development of benign pleural effusions. Something of an unusual side effect profile, which I think provides us a window into something about the biology of MET inhibition and potential resistance and safety. Next slide. One of the other things that we've realized with tepotinib and others, Becca Heiss presented this for capmatinib a few years ago, is that the kinetics for the development of peripheral edema are unusual. The median time to onset is between 2 to 3 months for the selective MET TKI. So it takes a while for this to ramp up, which is interesting. And the median time to resolution is actually quite long also, somewhere between 5 to 12 months, essentially. And another way to view that is that it really doesn't go away all that well. And so as a potential side effect, this, again, provides us a window for something interesting that's going on, which likely, given the kinetics of this, and anecdotally, I think for those of us who have seen it, tell us that something physiologic is happening in response to drug that we don't quite yet understand. Next slide. There are extant data, which I presented before, which I'm not going to present now, but I can refer you to the protocol and also other slide sets that have generated a hypothesis that dual VEGFR2 inhibition and MET inhibition can increase efficacy while decreasing toxicity. So there are an existing pool of data, particularly in non-small cell lung cancer, that selective MET inhibition ends up inducing upregulation of VEGFR2 dependency on VEGF-A as a ligand, and that this can be a way that resistance develops in a fashion that's not going to be captured by genomic analyses. So acquired resistance testing, generally speaking, has not really found a high frequency recurrent on-target or off-target pathway alteration to explain resistance. So the idea is that there may be something else going on that's just not captured genomically, and this we hypothesize is one of the issues. And the other part of this is that if this is happening in the tumor, we think this may happen in the periphery also. And there are mouse models for the development of lymphedema that would bear this out. And so inhibition of VEGFR2 or VEGF-A should lead to repression of peripheral edema as well. So hopefully within this context, improvements in efficacy, but also improvements, importantly, in tolerability for what is, at the end of the day, an elderly population of patients. Next slide. So this is the schema as it stands right now. Fairly straightforward, MET exon 14 skipping positive lung cancer patients, no prior angiogenesis inhibition, They have to be MET inhibitor naive, and right now they needed to have received prior standard first-line therapy. So again, a second line and beyond. Trial patients are randomized to tepotinib as a control arm or tepotinib and ramucirumab. We're looking for about 50 patients in total. Primary endpoint is overall response rate. Secondary endpoints are PFS, and then quite importantly, it's the safety signals that we're also looking for. We're looking for a decrease in all-grade edema, and again, as written now, lymphoscintigraphy scan changes to try to quantitate this as best as possible. Next slide. Right now, in terms of accrual, we opened formally in December 18, 2023, so several months after that, trying to get sites online. We do have 2 patients enrolled, one randomized to the control arm, one randomized to the experimental arm. And so I think that is something of an important milestone. I think the one who was randomized to the experimental arm was just about a month ago. But again, accrual, similar to, I think, Sarah's protocol, has been a little bit slower for reasons for us that are understandable. Next slide, which get to some of the things that we've done to try to improve the uptake of patients onto the protocol. One is that earlier this year, we released new resources for the study, reference materials for lymphoscintigraphy, since there were a number of questions coming up from different sites about this. So FAQs about that, FAQs for the protocol in general, and importantly, amendments. So there's one amendment that is in the works to make lymphoscintigraphy scans optional, to remove this as a potential barrier for sites. And so that will be the first amendment to get approved. And then for logistical reasons, we are then immediately going to follow that amendment up with another one that will remove the line of therapy restriction. So in other words, to convert this into a line agnostic protocol. So patients who are newly diagnosed, who are treatment naive, can go onto this protocol as would make sense, also matching the pivotal phase 2 studies as line agnostic protocols as well. And I anticipate that once, particularly number 2, is approved, that accrual should pick up, because ultimately NCCN guidelines, all of us have recommended trying to do this in the upfront setting as standard of care. Next slide. So that's it. Study contact, Juning and me, and medical questions is listed there. And folks have been doing a good job emailing us with different queries. And I think we've been doing a decent job trying to reply to them in a timely fashion as well. Thank you.
Thank you, Paul. So I will do my best to take the place of Dr. Gagiel today and talk about S1900J. And this is our newest open study at the end of September, just opened. And this, again, is a phase 2 trial of amivantamab subcutaneous in patients with recurrent or stage 4 non-small cell lung cancer with high MET amplification. And the study chair is actually Dr. Rolfo, who just moved over to Ohio State University and is working on transitioning all of his license and everything. And so will be the study chair with the co-chair of Dr. Gagiel and our other members, with Mary as our statistical chair, as always. So our schema is here, and as you see, it's MET amplification, and there are patients with high MET amplification. There's a non-squamous cohort of patients and squamous cohort of patients. Both are looking for 40 eligible, and the primary endpoint will be response by RECIST. And again, this is one of the first subcutaneous amivantamab studies. Just as a background, again, as we learned from the last study, MET is an oncogenic driver. Amplification of MET is a rare but also actionable oncogenic driver for patients with non-small cell lung cancer. Some were included in some of the MET studies, though there's less data available for MET amplification than for MET exon 14 skipping. I think it's around 3 to 4%. Most of the data is from non-squamous patients, and that's in part because that's who got most of the testing up front. And so I think this is also a novel trial in trying to understand this alteration in squamous cell patients. It can be targeted with TKIs, but also MET antibody inhibition. And so again, this is looking for at the antibody with EGFR and MET, both targets. So our population here are, again, in cohort 1, non-squamous non-small cell lung cancer. Mixed histology with any non-squamous component will go into cohort 1, and patients with squamous cell and will go into cohort 2. And response rate is our primary objective. And other objectives are overall survival, duration of response, frequency and severity of toxicities, and we'll be collecting cell-free DNA prior to treatment and at cycle 3, day 1, and at first progression. So this will help to understand this patient population. Just the treatment, again, the amivantamab sub-Q is a bispecific antibody that targets both EGFR and MET. We have found that this can lead to responses in patients with MET alterations. And just recently at ASCO, we learned that the subcutaneous version may have less toxicity and potentially even increased efficacy. And so we're waiting to learn more, but with this subcutaneous formulation, we do think that we will see less infusion-related reactions and have better tolerance. Because there are infusion-related reactions or administrative-related reactions, there are pre-medications and supportive care associated. This is all within the protocol, so I won't go into the details. But we do, again, some patients do have reactions, and some patients will need emergency medications too after. So here's the dosing schedule. They get a split dose and a loading dose for the first month, and then following the first month, it is days 1 and day 15. There are different dosing for sub-Q than there is for the IV, if you're used to using the IV, and there's different dosing based on weight with the 80 kilogram cut point. Again, the post-injection medications, our full eligibility criteria are here, but again, the main point is MET amplification. And this is currently by FMI, but will be...
No, actually, that's,
Okay.
so that was something I was going to bring up because, of course, what I have is Louise texting me with key information. If you don't know Louise, she's the data coordinator that runs everything. And yeah, so because MET amplification is a more complicated biomarker, MET amplification is only going to be based on a subset of the assays. And so there's a LungMap resources page that's online, so you can see, and on that page, there is a table, a study reference table that describes what the biomarkers are, because not all of the studies are going to just use the genomic alterations form. So if you do have a patient with MET amplification that you're considering for J, you're going to want to go onto that page to confirm that your patient could potentially come onto J, because it's not going to be just anything.
But it's not only FMI, so
Right.
It's not only foundation at this point. So it's probably best if you look at the resource page, and if you have any questions, to email the study team, because we are used to answering these questions, and I'm sure Sarah Goldberg has also answered a lot of questions about MET amplification in her study. So it is a little more complicated biomarker. I think the important piece of this study, again, in referencing S1900G, is that this is not for patients with MET amplification as a resistance mechanism. This is a study for MET amplification de novo up front. And so those patients who do have EGFR or any other oncogene driver, this is not the study for them. This is a directed for MET amplification, which we have many of these patients too. And again, the full listing, brain mets are allowed if they're asymptomatic. Prior therapies, I think, are pretty standard. All of these patients, the patients who have received prior chemo and immunotherapy are the majority of these patients, but there's not a lot of restrictions. They can't have had a MET TKI. And again, going back to the amivantamab, I think we covered most of that. Anticipated adverse events, again, many of them are EGFR-based, rash, diarrhea, and then the MET-based edema. So a lot of skin toxicities and the edema. Dose modifications, And we want to thank the whole team involved, and contacts are here. Thank you. And we will now have Dr. Cousino talk about the Translational Medicine Committee.
Thank you. So, what do we want to do in the Translational Medicine Committee? So, our priorities are to make discoveries, not just do descriptive analyses, although those are important too, to utilize lung map data and biospecimens from exceptional responders, but also from even the negative trials where we may not have seen differential outcomes, so that we can really learn from every patient whether the trial turns out to be positive or negative for its primary endpoints. We want to maximize utilization of the expertise in lung map, including pathology, immunology, and many other areas of excellence. We want to promote clarity and awareness of how to perform TM research in lung map. We want to garner support for this research, including outreach to advocacy groups. And we want to inform ultimately the next generation of lung map study concepts. So thank you for the shout out earlier about our manuscript, Mary. This is really the description of what we saw in S1400 in terms of the alterations seen in around 300 cancer-related genes on a research version of the Foundation One platform. There were 1,672 patients who, as lung map sees, had previously treated stage 4 or recurrent squamous cell cancers that were enrolled between 2014 and 2019. And so, these patients had an average of about 20 variants seen per patient, among these 313 genes. And as Mary indicated, this is, as far as we can see, the largest series of these squamous cell lung cancers in this previously treated space that exists. So there's a lot of data here that can be mined to see, what's the prevalence of different alterations or combination of these in this patient population that we're trying to address in lung map. But we tried to see if there's something novel in this data set, and this is, I think, the most interesting thing we found, which is they did these analyses to basically say which genes, when altered, are not seen with other particular genes also being altered. So said another way, if you have a KEAP1 mutation, you really don't tend to see either this NFV2L2 mutation, or it turns out this PARP4 alteration. Now, something that's been known for quite some time is that KEAP1 and NFV2L2, also known as NRF2, for people call it that, are two genes that work together to help repair certain kinds of DNA damage. And it's the kind of damage that needs to get repaired after people get things like platinum-based chemotherapy. So it kind of makes sense that cells who've been exposed to these kind of platinum-based chemotherapy kind of drugs might develop alterations in the genes that are related to dealing with such damage. But the surprise is that PARP4 was also mutually exclusive, did not have alterations when there were an alteration in one of these other two genes. So this is a clue more than anything, but that PARP4, which is a gene that we know practically nothing about, may have something to do with one of these pathways. And if so, it may be the reason why patients got resistance to their upfront therapies, and would be a new biomarker for that. Now, this is just a clue you could learn from sequencing data. My hope is that someone out there has a data set that's going to show that in a similar patient population, you also see these alterations in PARP4. And in fact, there are 3 hotspots here in particular where we saw dozens of mutations in these specific amino acids. And so it's the kind of thing where hopefully in time we'll learn new biology through the work we have here in LungMap. This is a busy table, but it's really meant more just to kind of show we're collecting lots of blood samples on LungMap. We're collecting eDTA plasma, Buffy coats, so you could look at the germline DNA, the patient's DNA. And since the LungMap 2.0, the LungMap protocol that's currently just been amended, we've been collecting ctDNA samples at multiple time points as well. Our committee recommended baseline, cycle 3, and at first progression. You can see that this was largely done in the majority of these sub-studies. What it's really looked at tumor circulation in blood to gauge responses in a very precise way. And this just gives a sense that we have hundreds and hundreds of these samples ready for your best ideas on how we could look at new biomarkers in the blood. Patients ask me all the time, isn't there a blood test for something like that? It's resources like this that will hopefully allow us to create such a thing. But then I want to just spend a couple of minutes talking about, now that we have LungMap 3.0, right, hot off the press, we're already working on what I might call LungMap 3.1, which is that, we've been looking at DNA all this time. We're going to start looking at RNA now. And if people remember from high school, you have DNA, it gets transcribed into RNA. That's how the cells decide which genes should be used to drive the biology of a cell. And then that RNA gets translated into protein. It's the protein that actually does the work, the enzymes, the structural elements of the cell, all that good stuff. And although we've learned a lot about how to treat lung cancer patients in a specific way by looking at DNA alterations, you're only going to find certain things only past the point of the DNA, but what the cell did with it, and so you'll only see it if you look at the RNA or the protein. So we're tackling the RNA already, as I mentioned, as of this amendment from this week. But we want to look at protein because there are things like antibody-drug conjugates. I'm sure most of you have heard about these, but these new types of drugs where you actually do need to know something about the expression levels of various proteins, especially the targets of some of these, or proteins that tend to be expressed in patients that may be more sensitive to these drugs. And since you have to look at the protein, we need to find a way to look at the protein. And then in immuno-oncology, yes, some of this is driven by tumor-related genetic factors, patient-related germline factors, but a lot of it is actually at the level of protein expression. And so if we want to understand primary resistance to these immunotherapy drugs or acquired resistance to these drugs, our group is very committed to trying to understand these states. We're going to have to understand this at the protein level. And so what's to come is how we do this in the way LungMap has done well. How do we screen for these sort of alterations? And so the current standard is if you're interested in a particular gene and how much of the cell is turning that into protein, you have to do immunohistochemistry, and you can only do this for one protein per slide. This is the time-honored way we look at these protein biomarkers, and it will be the way to really confirm whether protein really is expressed more than usual, let's say. But people have been working for several years now on how to do this on multiple proteins at once, even say 36 proteins at once, using variations on immunofluorescence or immunohistochemistry techniques, or using totally different technologies like mass spectroscopy. You also have to recognize that proteins can be altered even after they're made by all kinds of things that are added or subtracted, to the molecule that could drive how they work. And so these are all things that can only be looked at with some kind of protein-based screening. But, we're interested in a lot of proteins. And, the reality is if we ask for people to send in 50 unstained slides, we're not going to be able to get that very often because, these biopsies are just not amenable to that. And so we want to look at these proteins for these kinds of novel agents that we want to bring to LungMap, like antibody-drug conjugates, immunotherapy drugs. And so how we are doing that is we're working with companies right now to develop a technology that will let us screen. And this is just an example of a lead proposal where we may be able to, with just 2 slides, be able to look at 36 odd proteins. And so if we can do this, it's going to be a staged approach as we vet the technology and then start bringing it into LungMap. But we should hopefully be able to do what we've done for DNA, then for RNA, and now protein, find patients for the kinds of drugs that we are now wanting to test in our patients. So stay tuned. This is still going to be some months of development where we're working with these companies and finding the best solutions to do this. But this is, I think, what will get us to the next stage of our trials. And then will be a reason to continue being able to bring tissue to LungMap for testing that you can't get any other way. And I'll stop there just because of the time. There's more here, but I'm aware of the clock.
Thank you. It's great to see so many things going on with TM. And we will have Dr. Wakar talk about the DSC, and online we have Dr. Dragnev, who Dr. Wakar will become the vice chair of LungMap, and Dr. Dragnev will take over the role of DSC chair. So welcome both.
Thank you.
Thank you very much, Dr. Reckkamp. I also want to publicly thank Dr. Haas Borgai, Dr. Mary Redman, and Dr. Karen Reckkamp for their excellent leadership for LungMap. All the things that we've accomplished over the past year, it takes a village, but it also takes excellent leadership. So thank you for everything you all have done. And please, Jennifer Beeler and Gabe Chadwell, as well as Jennifer Newsom and Takwal Hussain, could you stand up so everyone can see the people who actually do all the hard work and project management behind the scenes? And also, Katie, can you stand up for statistics? Because really, they do such excellent work. And without them, there's no way we could do all the things that we do. So thank you very much for all of your hard work. Okay, so here are our disclosures. So the Drug Selection Committee is a group that basically comprises of many experts with representation from all the cooperative groups. We have a small internal group that meets to kind of really hone these study ideas, and the sources come from multiple areas. They can come from investigators, they can come from pharma companies as well. And then once we have a fleshed out protocol with statistical help from our statistics team, there is a process of formal voting with criteria that are used to see if this is a good study for a LungMap. Some of these studies fall into the biomarker-driven subtypes. Some of them fall into the non-match subtypes. And now that area is getting a little bit more gray as we're coming up with non-matches for biomarker-driven patients as well. In addition, we've also been working to develop immunotherapy studies, which are looking at biomarkers as well. So LungMap is going into a new era, and we're very excited to take it there. One thing that has been a bottleneck recently has been the internal drug selection committee meetings. And you can see that LungMap is a pretty quick process as far as getting the study moved forward. But it's really all the studies that we've been working on. Often the drugs have been earlier in development, and so the ideal time to engage with LungMap is when we have a dose that's been selected, and we know that that's the way to go forward, and we have some safety data available, and then we can generate the efficacy data through this sub-study. There's a lot of activity. We submitted these slides a month ago, and up till that point already we had 34 meetings just looking at this. And so we're very, very excited with all these concepts that we're developing and moving forward. Dr. Reckkamp talked a little bit about our retreat. Within that, when we discussed the drug selection committee and the role, some of the things that emerged were basically articulating better to our industry partners what is the benefit of working through LungMap? We have over 800 sites where the protocol can be open for the screening protocol. We already heard about the representativeness piece that's been presented and published. All these biospecimens that Dr. Kozano had talked about, the drug supply of this being through the industry, but the actual distribution being through CTEP. There's a path for regulatory approval as well, and then the competitive costs. We have been working on outreach to junior investigators and mentorship plan. Dr. David Gandera has really led a lot of those efforts. And as a result, I'm proud to say that some of the studies that are moving forward that will be coming through, they actually are led by junior investigators with senior investigators within LungMap serving as the co-chairs. And we're working on a formal mentorship plan moving forward as well. And then we have been engaging our translation medicine team very early within the concept development. Some of the non-matches that are coming through are going to have biomarkers that we're developing as well. And you already heard from Dr. Kozano about the other exciting things that are happening within the translational medicine team. And we also have novel studies looking at biomarker-selected patients, which are going to be kind of like the non-match after they've completed their targeted therapy and progressed on chemotherapy. I'm going to stop here, and in the true spirit of a hybrid meeting, I'm going to let our new chair for DSC, Dr. Dragnev, take over here for the developing studies update.
Thank you, Saima, and I hope you can hear me. I'll just present some illustrations of the activity of the Drug Selection Committee. I mean, all the studies that you heard earlier, they've gone through DSC. We have a protocol in development that hopefully will activate by the end of this calendar year. S1800E, it is a non-match study of docetaxel ramucirumab with or without an anti-PD-1 antibody. It follows S1800A. It enrolled very well, so we anticipate that this will be a trial that you will all open and offer to patients. Concepts in development is a long list. This is the most mature concept in development, S1900N, with a TROP2 ADC and a bispecific antibody against VEGF and PD-1. It is going to be a trial in patients with actionable genomic alterations. It is moving through DSC, hoping to activate the study at the end of next year. There are several concepts in various stages of development that I will be presenting at the next meeting. And with this, back to you, Saima.
Thank you very much. I'm going to let Dr. Reckamp do the closing remarks.
Well, thank you, everyone. I think we have just a few minutes. We do have some questions here. Yeah. And I do want to acknowledge Dr. David Gandera, who didn't have a speaking part, but is part of what started LungMap, and a part of a champion of some of these studies. So wanted to make sure, I don't know if you want to give a word, but. That's all right. Not like David to be at the end of the table and not speak at one of these. So he's letting the future of LungMap do the speaking today. And we have some questions from online, but if there are any questions in the room, we're happy to take them.
Yeah, so thanks for those of you who submitted questions online, and Louise is also online, so she's been addressing many questions, but I just wanted to address them for everybody to hear as well, and then of course, please interrupt if you have questions in the room. As I mentioned earlier, there is the study reference table online, and you'll want to look at that. Both S1900E and S1900J currently have that the biomarker is FMI only. S1900E is going to continue to have just FMI as the biomarker. When, as I was mentioned, the protocol amendment to reduce the sample sizes with CTEP, and then it'll need to go through the central IRB. And once we get full approval from CTEP to implement that protocol amendment, we'll give probably about a 2 weeks notice as to when we'll be closing that trial to accrual. So don't worry if you have a patient that you're working up, or even if you have somebody that you might be seeing in the next month or two, you have time to enroll that patient. And of course, we would actually love to over accrue. So if you have a patient, please let us know, and we would even potentially hold closure of the study if there's somebody coming on. There was a question about the revision 9 has a consent addendum. That consent addendum is related to the DNA and RNA testing for reanalysis. There are very few patients this consent addendum applies to. And those of you that consent addendum applies to you, Kevin Moraldo, one of the other amazing data coordinators on LungMap, has already directly contacted you. So if you haven't heard anything, the consent addendum does not apply to you, and you just are therefore required at some point within the next month to implement revision 9 and move forward with the new consent, but you don't need to worry about the consent addendum. In terms of, there was a question about pre-screening and tissue submission. So if you have a patient that you need to submit tissue for analysis, that is the same process as it is currently for LungMap. And what that means as well is that if your patient didn't have enough archival tissue to be submitted for on-study testing and you need to do a fresh tissue biopsy, LungMap will still pay for that biopsy. So there's no change there. We will still make certain that you don't have to try to find funds to pay for that. There was a question about the rationale for removing pre-screening for patients who are submitting a prior test, including those who are using prior FMI testing. As was alluded to earlier, basically we have seen that only about 13% of patients who pre-screen register to a sub-study. And because we're moving away from from this, where if you're submitting a test, you're going to get assigned a sub-study within a day of submission of that genomic alterations form. So there's really no benefit to either the study or the patient to go through all that it needs to be done if your patient is currently receiving therapy. But what we didn't talk about in enough detail here, and it's going to be online, David Kozno has developed this amazing resource for you to evaluate whatever test you have, and to be able to look at that test to figure out how you can fill out the genomics alteration form. In addition, if you have any questions about the test that you have on your patient and how to fill out the form, you can email us and ask us questions. In addition, for the first set of time while we're having this new change, our data coordinators, the committee led by David Kozno, will be reviewing the reports and evaluating if there are any key points of education that we need to be helping people understand how to translate this report into how to fill out the form. So don't worry, we are here to support you, and we understand that it is a major change, and sometimes those reports are hard to interpret. And so I think that I covered everything in terms of the online questions.
Thank you. I think we are at the end of our time here. If there are any burning questions, we're happy to answer them. Obviously, you can reach out to the LungMap team. But again, thank you to everyone for your participation and your ongoing support of LungMap. It is really an honor to be a part of this group that is, again, a decade-long study. Thank you.